Thank you very much, and thank you for the opportunity to be here as well. I've always wondered what the Colonnades Club is, so I'm the first time in 20 years to be here. So I want to tell you about our project, Realizing Increased Photosynthetic Efficiency. It's a 68 million investment from the Bill and Melinda Gates Foundation, the Foundation for Food and Agricultural Research, and also the UK Foreign and Development Office as well. It's an international project, so we're spread over 4 continents, but it is led by the University of Illinois. If I get the right changer. So just want to quickly tell you what the motivation is for engineering the process of photosynthesis. First of all, Illinois, for the last half century, has probably been the leading institution in the world for the study of photosynthesis. So fortunately, we were the go-to place for the Gates Foundation when they thought about the possibility of engineering this. The reason why we see this as important is we think about one of the biggest yield bumps the world has seen came from the Green Revolution, where we made genetic changes in crops, particularly in their ability to partition more of their biomass into the part we care about, the grain of wheat, the seed of soybean, and so on. And if we look at what genetically determines yield potential, i.e. what a crop can achieve at a given location from its genetics, it's determined by the product of 3 things. How good it is at intercepting the radiation that's available, how good it is at converting that radiation into biomass, and how good it is in partitioning that biomass into the part of the crop we really care about. And if we look pre-Green Revolution, we can see that partitioning into the grain, the efficiency of that was about 1/3 of what we think the potential would be. Today in our major crops, we're almost at 100%. You know, wheat, for example, 60% of the biomass ends up in the grain. So really isn't much room left for further improvement. Interception efficiency has also improved, but conversion efficiency, which is the process of photosynthesis, has been hardly changed. So why do we think we could do something about that now? Well, because we understand the process in great detail. We can simulate the whole process computationally, work out where we need to intervene. And of course, with gene editing, genetic transformation, we can also attack the process in a way that wasn't possible before. The way we work is, first of all, to predict from computer simulation where we should try and intervene in the process, and then try and do that. We do that, first of all, in tobacco, which will sound very odd to you. The reason for this is that tobacco is very easily modified, and it produces a lot of seed. So we can go from genetic transformation to trial in just 12 months. So we use that to really filter, okay, does our computational idea really work in practice? If it does, then we go on to the much harder crops of cowpea, black-eyed pea that is, cassava, soybean, and also maize. And so we've had some successes in tobacco, and indeed we've now managed to transfer one of these to soybean, and in field trials on the farm, that showed about a 20% improvement. So we're pretty excited by those results, and the Gates Foundation is now discussing with us going into a 3rd phase. But the other thing they've also done is to fund on the research park 2 new facilities which are allowing us to accelerate this movement of our successes in tobacco into food crops. And the first of these is the crop transformation facility. This is a state-of-the-art facility which allows us to generate our constructs robotically, then put those into soybean, which is our major focus on there, but there's also work on cassava and maize as well. Of course, when you're generating all these plants, you want to know quickly if your transformation is making a difference. And so the other facility that we've now built on the research park is the high throughput phenotyping facility, which some of you may have seen this, what looks like a large greenhouse, which has been developed there. It's using geothermal wells to cut down on all the costs of heating and cooling of that. But in this facility, each of 400 plants is sat on a load cell, so we can monitor immediately how much water that plant is using every day, how much it's growing. And we also have above there sensors which are also looking at the development of the canopy. Now, if things are successful in here, we then move them forward into the field. And some of you may have seen this. It's by Race Street and Curtis. And there are these 4 pylons, which are about 150 foot high. And strung on those is this thing we call spider cam. So if you've been at the Bears Stadium, for example, you see this camera whizzing across the field. Well, this is exactly the same technology, except hung on here, we have a series of sensors which are monitoring things like the growth of each individual plant over a 10-acre plot, and also some of the photosynthetic characteristics. So with that, I just mention our most important, well, I should mention that we've had quite a lot of media attention, and in fact, on BBC World News this Saturday, this phenotyping will actually be featured. BBC has a series called Follow the Food, extremely popular in Europe, but it's now having an impact in the United States as well. We were also featured for a large chunk of a PBS Nova program earlier this year. And we also had a large feature in the New Yorker magazine. I should just mention our most important facility, though, is the team. And we've been really lucky to assemble such a dedicated team of colleagues who have really made all of this possible. Should also mention that the Gates Foundation has established a new unit which has funded these facilities on the research park. It's based in St. Louis, called Gates Agricultural Innovations. And their job is really to take our inventions and actually get them into seed systems for some of the poorest farmers in the world. If you want to know more, we have a website, very simple, ripe.illinois.edu. Thank you. Is there any time for questions?
We have questions. We have time for a few questions if anybody would like to know more.
Yeah. It's not on, yeah. I could say, I can repeat the question. Yeah, so the question is about carbon neutrality and what this project might do for it. Clearly the motivation of the Gates, primary motivation of the Gates Foundation is to get more productive crops into the poorest countries in the world. But of course, whenever you increase the productivity of a crop, there is also going to be, if you like, more trash on the field. There's going to be bigger root systems. So you are adding your potential to add carbon to the soil goes up. But another possibility we're investigating through the US Department of Energy with these technologies is, well, in the West, if we want to start thinking about putting more carbon into root systems, if you have more photosynthesis, you are assimilating more carbon. So you could put more carbon into, if you like, more less labile parts of the plant, which would stay on the field without reducing yield. And,
Yeah, here we go. I think Jim Lenz has a question over here.
So when I think about evolution and the efficiency of photosynthesis, I'm wondering, why do you think nature didn't achieve what you're trying to achieve?
Yeah, that's a great question, and obviously we've thought a lot about it. And we have some of the answers, not all of them. But of course, one thing is, if we think of our crop ancestors, evolution is about getting your genes into the, already genes getting themselves propagated into the next generation. And there are some very successful plants which are hugely unproductive, but they're just very good at dispersing their seed. So productivity is not the primary selective factor under evolution. Two other changes is, we found, for example, that many of our crops, photosynthesis poor in shade conditions. Now, if we look at, like the ancestor of soybean, it was a climbing plant where nearly all the leaves are in sunlight. Today, we're growing it in dense canopies where 75% of it is in shade. And so photosynthesis in shade in soybean is quite inefficient, and that's something we can address as well. Most of our crop ancestors were actually sun plants. Today, we're growing them in shade. The third factor that we found was we found that we could, if we upregulated an enzyme, which was really a very small component of the protein of the plant, we got an increase in photosynthesis. And we puzzled about this. So we then went back and we ran our model of pre-industrial CO2 concentrations. This enzyme had no effect. But when we ran it at today's concentrations, this enzyme was a bottleneck. And CO2 has more or less doubled since the start of the Industrial Revolution. Half of that increase has occurred in my lifetime, which is very few generations, really, for a crop to actually adapt to that change. So I hope that's a partial answer to a really challenging question.
Thank you. Do we have other questions in the room? I'll get you a microphone. While I look for hands, I'm going to see if Steve can tell us a little bit about, this improvement in photosynthesis is made possible by a lot of the biology, crop science work, but it's also computing. And I think NCSA, the National Center for Supercomputing Applications, has had a role in enabling these advancements. Can you talk to us about the intersection of those two factors that are important here?
Sure. We're lucky in that the process of photosynthesis, just about every step in that process is known. We know all of the genes involved. We know the protein products. We know a lot about their kinetic properties. So what this allowed us to do with NCSA was literally simulate the whole process. So basically we could, every discrete step from capture of photons to conversion of carbon dioxide into sugars and starch, we could represent as a system of differential equations, which you could then numerically integrate. To do that, you need a supercomputer, which was perfect for Illinois because we had one. So what we could then do was to simulate the whole thing and then start changing each of these proteins in turn. And of course, 99 times out of 100, you just make it worse. But one of those, it actually makes it better. And so that was what we were homing in on. Out of all those things you could change, which are the ones which might actually make it better? And then with transformation, we could then see, well, does this work in practice? And we've now had 3 of those which we've been able to demonstrate. I should say, before we could do that, there was a lot of skepticism, as you were saying, well, surely evolution would have already done this, or breeder selection would have already done this. And so this has really changed minds, because we've shown 3 very different ways we could actually achieve higher photosynthetic efficiency. And in tobacco, each of those has given us about a 20 to 30% improvement in productivity. One of those has now worked in soybean, another one has worked in potato. But we're moving all of these to soybean and to cowpea. So, okay, thanks.
Oh, Manfredo's got a question. We'll do one more before break.
Yeah.
Hi, Steve. Just a question. Plants are within the environment. Photosynthesis depends on many factors in the environment, like fertilization, water, and now microbiota, that they are able to regulate genes in the plant and affect photosynthesis. Mm-hmm. So if you can tell us how the project can, how integrate all these factors to have an impact on the photosynthetic efficiency that you are doing?
Yeah, I mean, we don't know how environment is necessarily going to affect that. I mean, we do know, whenever we improve yield potential, that it isn't just in the best environments where we get an improvement in yield. You can see this with soybean and with wheat, for example, historically, if you look at that. We are, now with the soybean, we are doing multi-location field trials, so across a range of environments, to really see, can we get this yield increase in a range of environments? How does drought affect it, etc. Obviously with soybean, we're not adding nitrogen to those, so it's M-fixing. One of the exciting things we saw on the trials here was that although we got over a 20% increase in yield, the protein content of the seed was not affected. So we assumed the plant must be using some of the extra photosynthesis to feed the bradyrhizobia to get that nitrogen. Some of the traits we're looking at as well give us more photosynthesis without costing more water. So we are, at least in one case, making the plant more water use efficient, which we think will be beneficial in lower water environments.